This video will be showcasing the design, process, and findings of the Tatsoi aeroponic grow study. Dr. Kuwahara requested this study on Tatsoi to determine the best way to grow the crop using aeroponics, and to learn about its characteristics when grown in an indoor environment. In this project, we were first required to thoroughly research Tatsoi. We needed to initially know how long to germinate the seeds, how quickly the plants grow, and what their ideal environment is. Then, we had to design and carry out a study based around this research. We were limited in time and growth space and took these constraints into account when designing the plan. The grow study took place in the aeroponic systems in Shop 4 and was finished on November 10th. The aim of the study was to collect data for indoor farmers so they have more available information on proper harvest times and optimum seed planting per slot in order to produce the largest and highest quality harvest of Tatsoi. The people who worked on this project are Anna Rio, Alec Bergerman, and Chloe Kuntz.
This is the A Essence aeroponic unit. That is the water basin. These are the different grow tubs that the plants lay in. These are the nozzles where the water and nutrient comes out to spray the roots of the plants. These are our different lights. Both the lights and the spray rate can be controlled in an app. Our lights were turned on for 18 hours and off for 6 every day, while our spray rate was for 15 seconds every 5 minutes, which was later changed due to water issues. Before we could start the planting process, we first had to clean out the planter. This first photo shows us filling up the water to figure out how many gallons it would hold. The second image shows Chloe cleaning out all the leftover salt from a previous experiment. Next, we had to take out each tub, clean it out, and then also reassemble. Alec is standing next to an unassembled aeroponic unit.
Next, we started seed germination. First, the floor foam cubes were washed twice with water and left to sit in a 10% hydrogen peroxide and 90% water mixture in a tub. There were 99 cubes total. 33 contained 1 seed each, 33 contained 2 seeds each, and 33 contained 3 seeds each. As you can see in this picture, we are placing the seeds into the cubes. The cubes were then left in darkness on a heating pad for 2 days, and then were uncovered and exposed to light on the third and final day of germination. In this picture, you can see the covering over the seeds.
After germinating, the next part was making the fertilizer. We have 3 different parts of fertilizer. Part A, which had nutrients, Part B, which was also nutrients but more of a nitrogen base, and Part E, which was an Epsom salt water solution. We had to have 3 different parts and add them at equal portions in the order Part A, Part E, and Part B, while stirring in between each one, making sure that the solution was perfectly stirred. In this video, Alec is filling up our water basin to the marked line, where we have 30 gallons marked. We must do this every time we have to harvest, because the plants would take water and the nutrients out, which means that we'd have to refill it up. Right there you can see our equal parts of fertilizer. Alec is adding fertilizer to the water, while Chloe is using a yardstick to mix it in.
To harvest tatsoi, we first remove the plants from the aeroponic grower and then separate the leaves and stems from the roots because we only want to measure the part that will be eaten. This process is demonstrated here on a larger plant. Next, we calibrate and weigh on the scale, which is measured in grams. To summarize, we completed an aeroponic grow study on the tatsoi crop, which tested for the optimum grow time, measured in weeks, and the optimum seed density that created the greatest harvest. We found that the best time to grow tatsoi is 3 weeks or 21 days, and the optimum seed density is 3 seeds per growing slot. The main problem we encountered was most of our plants dying shortly after being planted in the aeroponic unit because the sprayers did not reach them. We addressed this issue by increasing the spray rate and spray time to make up for the water barely hitting many of the plants. This helped, but many of them still died. There are a few possible design improvements that could address this issue. One option is to germinate the plants for longer until they grow long roots that can reach the water being sprayed lower down in the machine. An alternative is to use only the bottom 2 layers of the aeroponic unit. As we found, when the water is shut off to the top layer, the water pressure increases to the bottom 2 layers enough so that the sprayers can reach all of the plants. But this obviously sacrifices 1/3 of the capacity of the machine, so finding other ways to increase water pressure is highly recommended. Another improvement to the project is to closely monitor the water and nutrients levels every 2 to 3 days. The water level, nutrients, and pH depleted faster than our group anticipated, and we were not able to correct these until they had reached quite poor levels. Keeping the nutrients and pH at appropriate levels is especially important in a highly controlled system like aeroponics, where without the buffer of soil, changes to water quality affect the plants directly and quickly. A final improvement to the design of this study is to measure the growth of the tatsoi over a broader time period, as growing tatsoi for longer times, such as 4 or even 5 weeks, could reveal useful results.
Here's a quick look at our graphs. The highlighted portions are the best grown plants from each section.